Nolan Williams. I'll be speaking briefly about our technology we developed in the lab, which we called SAINT, or Stanford Accelerated Intelligent Neuromodulation Therapy. SAINT is a stimulation approach. We currently apply this to a transcranial magnetic stimulation device, and the idea is that it's a kind of rethinking of how to do brain stimulation in psychiatry and in potentially neurological illness as well, where you're thinking about the where, and the where meaning kind of the personalized brain network target, the how meaning how do you stimulate. In this case, we use hippocampal physiology, mammalian physiology to guide that question, and then the how much. And so there's clearly a dose response that can happen in brain stimulation, similar to how it works in pharmacology as well. This is another way of looking at that same information. And so where in the brain that you are stimulating is up on the left A panel, and it's different in everyone. And then the kind of how is the right panel. So using space learning theory based stimulation, so playing a memory signal into the brain to tell the brain to turn on, remember to turn on and stay on. And then this C panel showing the how much stimulation do you need in order to have the behavioral response that you intend to get. And that's different in everybody as well. So SAINT really takes into account those three fundamental questions. And so we give 1,800 pulses of this hippocampal physiology-based stimulation approach, and we're able to compress an entire 6-week course of theta burst TMS into a single day. And we do that in an individualized, personalized medicine target. So our randomized controlled trial that we published in the American Journal of Psychiatry a couple of years back, these were 22 to 80-year-olds with a MADRS and HAM-D score in the moderate severity range or above, and treatment resistance at the kind of moderate to high treatment resistance level on the Maudsley refractoriness scale. And what we found is that, we were able to recruit, very severe folks, an average length of current illness of around 9 years. 50% of people were on disability from depression, 5 plus or minus 2 med failures. And they had a moderate to high severity on the depression acute severity scores, suggesting this was a pretty sick population. And we found, pretty striking response rates on the bottom. You can see this, kind of 70-ish and above percent response rates, and Cohen's d effect sizes above 1 any way that you analyze this data, which is a very large effect. And you can see that most people in this cohort crossed the remission line at some point in the 4-week follow-up in the active group, whereas in the sham group, a minority of people crossed that remission line. And then we found a biomarker where we can actually predict this effect, and this was published in Proceedings of the National Academy of Sciences. So we actually looked at the time series of the blood fluctuations in these brain regions that reflect electrical activity. And in normal healthy controls, this dorsolateral prefrontal cortex precedes the cingulate kind of conflict sadness brain region. And in individuals receiving SAINT, we were able to flip that signal to make them look like normal healthy controls after you treat them on the post-scan. And that flipping of the signal correlated with changes in the MADRS score if you measured it in the anterior cingulate. But in the pre-treatment, what we found was that the pre-treatment depressed individuals, majority of them had the signal flipped so that the cingulate preceded the dorsolateral prefrontal cortex. And that was true in our scanner. That was replicated in the WashU scanners. And the folks that had the abnormality are the ones that remitted, and the ones, and the ones that were able to show a clinically relevant response to SAINT, whereas the people that were non-responders to SAINT had a normal looking brain from this perspective. and the severity, as you increase the severity, the probability of the signal being there goes up, suggesting that we have early makings of a biomarker prediction for SANE. So we're really excited about that. The big takeaway is, we can use neuromodulation to get people out of, very severe chronic depression, an average of 2.6 days, and from our perspective, that's very important because it matches the speed of treatment matches the acuity of the illness. So this whole approach that I'm talking about is FDA cleared, breakthrough status, and now paid for by Medicare, so paid for through innovation funds for inpatient and outpatient hospitals. So SANE is something that people can get and implement into their clinical practice right now if they treat Medicare patients, and hoping that commercial payers will follow. So it's not something that, is a future aspiration. It's something that can be done today.